All right. All right. So today is day two of mitosis, and today what we'll be talking about is DNA replication, asexual reproduction. Um, and so remember, DNA replication that occurs inside of the S stage of interphase. That's what we talked about last class. And today we're actually going to get into more specifics on how that happens. We'll also have an activity that talks about replicating a DNA strand and what that looks like as well. After that, we'll go into asexual reproduction. And so I just want to talk about this vocabulary term real quick. What does it mean to be asexual? So anytime that the prefix a or an is put in front of something, it means not. So this kind of goes back to this whole the science, the Latin origin, and Greek origins on terminology. And so whenever we say that it is asexual, we're saying that it is not a sexual form of reproduction. And sexual reproduction involves two individuals. Asexual reproduction only deals with one individual. And also, whenever you have asexual reproduction, it's always going to be identical. Whereas in sexual reproduction, it's not going to be identical to the parents. And that's what we're talking about, identical to the parents. That's not going to happen. All right. So today we're going to talk about asexual. Whenever we do talk about sexual, that will be in our unit on meiosis. DNA replication. So, of course, like I said, this occurs during the S phase of the chromosomes, where the chromosomes replicate, where the DNA itself, the DNA strands are replicated. The amount of DNA in the cell is actually going to be doubled. And the two new strands of DNA contain one old strand and one new strand. That's actually going to be a very specific term that we call. And what that term is, it's called semi-conservative. That means that we kept one of the original strands, but we paired it with a new strand, just like we on the other new strand, we have an original and a new with it. And this image, I think, does a great job of depicting semi-conservative. So at the top of this image, we see the one DNA strand or the one DNA molecule where we see that double stranded there, and then we start to see that it starts to split apart. But as it splits apart, we start adding in new nucleotides, and those nucleotides match with their base pair, which we're going to get into in a second. And then that is what creates the two new strands. One having an original and each one having an original and new. And that's what's meant by semi-conservative. So DNA is made up of a double helix of complementary strands. Whenever we say that it's complementary, that means we're saying that the two sides go together. Fit together like a lock and key. During replication, these strands are separated. Now, whenever we talk about these strands going together, what we're talking about are those nitrogen bases. So if we could think back on whenever we talked about a DNA molecule being a nucleic acid, we said that each DNA molecule was made up of a phosphate group and a sugar group. That's those ribbons. But then the middle of that ladder are the nitrogen bases. Well, right here, they pair together, and this is what we mean by complementary. What two go together? So A pairs with T, and C pairs with G. All right? Each strand of the original DNA molecule serves as the template for the assembly of its complementary new strand. Okay? So that means if on the original strand, if I have an A present, then I have to attach a T to it. If the next nucleotide is a C, then I have to attach a G. If the next one's a T, then I have to attach an A. And that's what we mean by serves as a template. And during our activity on this, you'll know exactly what this is because you will be doing this. And it'll make it a lot more understandable for you. So during this process, that the DNA strand doesn't just separate on its own. It actually has to have enzymes. If you remember, whenever we talked about enzymes in our second unit, I told you that if it ends in ASE, it's an enzyme. Helicase is an enzyme, and so is the second one down here, DNA polymerase. Now, DNA helicase, it's an enzyme that's going to untwist it and unzip it. And the reason why we say unzip is it kind of looks like a zipper where you have the two sides connected, but then they break apart. And so helicase is the enzyme that's going to untwist and unzip the DNA strand. Right here where it says DNA polymer, polymer is talking about, making it really long, making it big. So it's making. So this is one that's going to make, it's going to take the new nucleotides and it's going to add it in. It's going to add those complementary nucleotides in. So like I was saying on the last slide, if there's an A, we're going to put a T there. If the original strand had a C, we're going to put a G there. And so that's the job of the DNA polymerase. But the job of the helicase is to untwist it, to straighten it out, and to unzip it. So whenever we talk about chromosomes, a lot of times I've had students in the past get confused on what's the difference between DNA and a chromosome. They're the same thing. All DNA, so all a chromosome is whenever we tightly coil up DNA, it's going to form chromosomes. Okay? And we really only do that to get ready for division. I'm sorry, there you go. We really only do that at that division stage whenever we're just about getting ready for prophase to start happening. All right? And that's what we're going to get into next class is actually what's happening during that division of a cell. So the duplicated chromosomes are called sister chromatids and are held together by a centromere. So inside of it, which technically it's not inside of it, it's not like it's stuffed inside of it, but for the sake of right now in your level of education, it's okay to think about it that way, but that's not really how it is. They're really just all coiled up and tightly bunched together and braided together. Well, we have our two copies of that DNA. Remember, we made an identical copy. Well, on one sister chromatid, that has one of the copies, and on the other sister chromatid has the other copy of the DNA. And remember, those two DNA strands are identical to each other. And whenever we get into mitosis, we're actually going to see why this has to happen this way in order for both daughter cells to receive, because they're each going to receive one of those chromatids, and boom, that means they now both have the same DNA inside of them. So asexual reproduction. So like I said, asexual reproduction involves a single parent cell dividing to make two new identical daughter cells, or it could also involve just a single organism dividing to make two identical, or that will end up being two identical organisms. So examples of this is mitosis, of course. That's what this whole unit's about, is in general mitosis. We also have binary fission, budding, vegetative propagation, and regeneration. Now, I also put on here lizard tails. There's also, so that would be regeneration. Technically, what starfish do is fragmentation, and I have that on the slide that we're going to cover. But really, they're so similar that I just went ahead and put them together on one slide to where it's broken apart individually. So what is mitosis? Mitosis is things like for our skin cell replacement. If you think of your skin cells like shingles on a roof, that's how they're layered. But you constantly get rid of dead skin. Kind of like whenever you get a sunburn and your skin peels, where you're peeling off skin cells, and so you're going to have to repair those. You're going to have to remake new skin cells. You also have growth and development, just for regular getting bigger. You're not the same size as you were whenever you were a baby. So you had to grow and develop. It's also for replacement. So if you cut yourself or repair. And this happens in somatic cells. Your somatic cells are your cells that undergo mitosis, such as your skin, your bone. Those undergo mitosis. The chromosome number is the same. Remember, they're identical. But the daughter cells, they're just smaller because they haven't grown yet. So that's why I have this right here. I have the term diploid. That's what diploid means. Diploid is talking about a complete copy of the chromosomes. And we give that the letter 2N, the N meaning the number of chromosomes. And the parent, and so that's the parent cell there on the left. And then it's divided into two daughter cells at the end of mitosis. And those are, of course, are the same. So you're going to see some images that could easily look like this on your quizzes and on your tests. You're going to need to make sure you know that. A common mistake that people will make on this image is they'll think that the 2N is divided and each one of these individual cells, these daughter cells, will have a 1N in it, thinking that 1 plus 1 is 2. That's not the case. Remember, these in mitosis has to be just like the parent cell. So if it says 2N in the parent cell, the two daughter cells also have to have 2N in it. Remember, identical in every way. These are just a little bit smaller because they haven't grown yet. Binary fission. Binary fission is nothing more than how a prokaryote, a bacteria, divides. And remember, it's identical. It's a form of asexual reproduction. You won't have to know all the steps to this. What you'll need to make sure you know here is you'll need to know that binary fission is a type of asexual reproduction. You'll need to know what asexual reproduction means. That means that the new bacteria will be identical to the original, and that this only occurs, or this is occurring. I'm not going to say only. This is occurring in prokaryotes, and remember, prokaryotes are bacteria. The next one we have is budding. This is when a new organism develops from an outgrowth, or we can just call it a bud, at a particular site. So if you see this one, if you follow the picture order, in this first image, there's nothing there. We get a little bit of a bump maybe. Second one, we start having that bud, that outgrowth. And the next thing you know, it just keeps happening. And boom, it's smaller, then it grows into an identical as the original. So that would be budding. Regeneration and fragmentation. So regeneration is basically you regrow something that would be like a lost part, such as a lizard regrowing the tail. Fragmentation is the ability for each severed section to grow into a new organism. Now, I'm going to say something that's it's not completely accurate all, because technically each piece has to have all the cells and everything required to meet, for that organism to be able to live. But let's say the starfish. I'm just going to use a very general example. If we cut off the spine of every single one of these this starfish at the very beginning, and we cut off all 5 spines of it, then that means we're going to have 6 starfish at the end of it, because each spine will grow a new starfish, and that middle part will grow a new starfish. So that would be fragmentation. The pieces would grow into a new one. Okay, and hopefully that example with the starfish really help out. Next one is vegetative propagation. Don't get this confused with like pollination and that type, because that's not what this is. Because pollination would be sexual reproduction. Vegetative propagation is when a new plant is created from a part or a clipping of the original plant. This includes the original plant stems, even the leaves, and even the roots. Basically, weeds will do this. Strawberries will do this. A lot. Kudzu does this. Really, a lot of plants will do this. So basically, there are a lot of bushes out there and flowers that my mother grows that if you go and take a leaf off of it, you go put it in water and make sure it has its nutrients and sunlight, it'll actually start sprouting roots and it'll become that new plant again. And it's nothing more than a clipping. So that's what vegetative propagation is. So day 2's review, I know it might seem like a lot, but it really wasn't that much. Explain the process of how the DNA strand is replicated. Don't forget about those 2 enzymes. So basically what we have to do, the process, step one, is we're going to have to take the DNA, we're going to have to untwist and unzip it. That would be helicase. And then we have to have DNA polymerase to add in the new bases. And that's going to give us two new identical strands or two new identical DNA molecules. What nitrogen bases are found in DNA? That's nitrogen bases A, T, C, and G. The A stands for adenine, the T, thymine, the C, cytosine, and the G, guanine. How many chromosomes are in a typical human body cell? 46. What is asexual reproduction? When the parent cell or the parent produces an identical copy of itself. In asexual reproduction, how do the offspring compare to the parent cell? They're identical to the parent cell and they are identical to each other. And what are the different types of asexual reproduction? And be able to identify them. Now, I might give you the definition. I might give you a written prompt to where you have to figure it out, or I might give you an image to help with that. So make sure you can do each one here. Remember, we have mitosis, binary fission. We have vegetative propagation. We have budding. We have fragmentation and regeneration. So make sure you can look back at it. And also don't forget those DNA strands. They're considered semi-conservative because each strand contains one of the original that was the template for the new one that it was made off of. Okay? So next class, what we're going to be doing is going over mitosis specifically. We're going to be going over the stages of mitosis and how that occurs and what occurs during those stages.